We can understand the concept of mitosis through drawings easily, and usually a normal parent cell is in diploid state, which means the parent cell has 2 copies of each chromosome. Okay, let's look at this first pair of chromosome that represent the chromosome that have the same shape, same size, and carry the same genes. Okay, because this is diploid parent cell, so this chromosome will exist in 2 copies, and usually we use white color for one of it and another one we color with pencil to make it dark color. Why? This is because one of the chromosome is come from maternal, inherited from mother, and another one inherited from father, or we say paternal origin. Another pair with another pair of chromosome. This one, they are actually same size, same shape, and carry the same genes. Again, they always represented in 2 different colors to imply the different sources of this chromosome. Okay, the white color, or usually in reference book, they will be colored in red. The red or now is white color chromosome. They are inherited from mother, maternal origin. And the darker color or in theory book, you will notice they usually will be colored in blue color. Okay, it will be represent the chromosome inherited from father, or we say paternal origin. So you will notice in this chromosome, half of the chromosome will be inherited from 2 different sources. So how many chromosomes in this cell? So how to determine the number of chromosomes? Usually I will guide students to look at the number of centromere. So here is 1 centromere, 2 centromere here, even though it's dark, but we still can identify here got 1 centromere, number 3 and number 4. Okay, so this is a shortcut, okay, help us to determine the number of chromosomes. If you there are 4 chromosomes in this cell, and usually it will be written in this formula, which is 2n equal to 4. The 2n is read as diploid, which means the polity status of the cell is diploid cell, because each chromosome will be found in 2 copies. As I said just now, this is a pair of chromosomes which are same size, same shape, and they carry the same genes. Just they are inherited from different origins. One from mother, another one from father. Same goes to another pair of chromosomes. They are actually same size, same shape, and carry the same genes. So 2n equal to 4 means this is a parent cell which is diploid cell, and there are 4 chromosomes found in the nucleus. Okay, let's start with the first stage of mitosis, which is prophase. And if we are given a parent cell with diploid 2 chromosomes, that means we have to draw 2 chromosomes only in this parent cell. And the 2 chromosomes actually refer to same type. Because why? This is a diploid cell. Diploid cell has the meaning that each chromosome, they exist in 2 copies. Okay, so what can be implied in the drawing is, we remember that prophase, the nucleus is still observable, but nuclear envelope start to disintegrate. So the disintegrating nuclear envelope usually will be drawn with dashed lines. And the disintegrating nucleus, I will use dot to form a spherical pictures to represent the disintegrating nucleus. Okay, now we focus on the chromosome. There are 2 chromosomes in this parent cell. So I draw the first chromosome, okay, which is duplicated chromosome, or the chromosome have two sister chromatids, because this cell is already undergone the interphase, okay, which include the S phase. And this is diploid cell, so each type of chromosome will be found in two copies. So try to draw as similar as it can, okay, same size, same shape, but actually they carry the same genes. Just I'm able to list down all the genes clearly in this schematic diagram. And I usually will practice one is in darker color using pencil, okay, another one is remain in white color, because the diploid cells actually means that they inherit the genetic material from their parents. So half of the set of chromosome is inherited from parents. Okay, so after we have complete the drawings of the nucleus, we have to complete also some structure which is significant in the cytoplasm, such as the centrioles that can be found in animal cells. So it can be found in a pair which arrange perpendicular in the centrosome, and there are two centrosomes at the prophase. And they start to separate because of the elongation of microtubules between the centrosome. So they push each other away. Okay, so this is the drawings of prophase. So must try to label, yeah, must at least give 3 labelings. So I always label the most important structure to guide us in study the mitosis, which is the chromosome here. And also you can label the structure such as nucleus, okay, and also the nuclear envelope. Both start to disintegrate here. And also the spindle fibers, okay, which is actually the elongated microtubules. Or you can label the centrosome, or even the centrioles, which is only found in the animal cells. The second stage of mitosis is metaphase, and usually it is characterized with a metaphase plate that is located at the equator of cell. Okay, we know that this is not a physical structure can be found in the cell. Okay, it's just a plan, a location where the chromosome will align or arrange at the metaphase plate. Okay, since that there are no more nuclear envelopes and nucleus found in the cytoplasm, okay, this is why now the chromosome can show the different behavior after the prophase. So just now we have the parent cell with diploid with 2 chromosomes, right? So our story must show the continuity, mesti ada kesinambungan. Okay, so I draw the 2 chromosomes align at the metaphase plate. Okay, because just now I draw one of it with darker color. Okay, so this one I also draw with the darker color. One of it here is still 2 chromosomes. Okay, but how they can be arranged at the metaphase plate is actually the function of the spindle fibers. Okay, where the spindle fibers is actually developed from the centrosome. Okay, or we say it's organized by the centrioles. Okay, so we have the centrioles or the centrosomes now is already located at the opposite poles of cell. Okay, dia memang berada di jarak paling jauh dalam satu sel. Okay, opposite poles of cell. And each centromere of the chromosome will be attached with the spindle fibers from the opposite poles of cell. Okay, so same goes to this one. The spindle fibers will elongate and then attached to the centromere. So one centromere will be attached to the spindle fibers from both poles of the cells. Okay, this is the spindle fibers. Okay, they have the name called as kinetochore. Okay, kinetochore microtubules. Okay, because they will attach to the kinetochore at the centromere, or we can just use the general term spindle fibers. Okay, some of the spindle fibers, they are elongate and slightly get across the metaphase plate, but they does not attach to any centromere. Okay, this is the one from another pole. So this is the non-kinetochore microtubules, okay, which is not attached, okay, to any. So each drawing we try to remember 3 labelings. So now the kinetochore microtubules or spindle fiber is one of it. The second one, I still encourage we focus on the structure, which is the most important in this topic here, how the chromosome, okay, show their behavior. Okay, so this is chromosome, which now is already arranged at the metaphase plate. And okay, again we can label the centrosome. Okay, or you want to focus on the centromes. So this is the drawings of metaphase. When move on to anaphase, the animal cell will start to show changes in the shape, which now they are found in spindle shape. Okay, this is because the elongation of the microtubules made the cell slightly elongated. So we always remember that the centros or the centrosome, they are always located at the furthest distance in a cell. Okay. Macam couple yang telah pisah. Okay, so this is wrong if we draw the centros or the centrosome like this. Okay, this is incorrect because it's impossible to be found in this distance. Okay, even though students say this is also opposite poles of cell. No. Okay, we must always remember the opposite poles of cell means the longest distance for the location, for the position of the centrosomes. Okay, so just now we know that the metaphase is already arranged at the metaphase plate. In anaphase, we don't need to label the metaphase plate anymore because why? Okay, the chromosomal behavior of chromosomes start to show the keyword here, which is separate and move. Okay, so I draw the first chromosome upper is the plant color chromosome, and then their sister chromatic also found at the opposite poles. Okay, the picture maybe is not the best one. Okay, I redraw because we try to make it as similar as it can here. Okay, so I try to draw one more time. Just now because myself, the white color is located at the top, right? So this one I try to make it same as it can to show the continuity, okay, kesinambungan cerita itu. So we have the another chromosomes, the darker color one, okay, whereas the sister chromatics separate and move. The reason is because the centromere split. Okay, the centromere split, and then the shortening of the microtubules, okay, cause the separation and the movement of sister chromatics apart to each other. Okay, and always remember when we draw the sister chromatics, the separated sister chromatics, let the centromere face towards the centrosome. What is mean here, if student draw in this way, right? If let's say you draw like this, where the microtubules is here and here is the centrosome. This is the wrong drawings also. Okay, same goes to this one, the separated pair. If you draw, okay, the tips of the chromosome arm face toward the centrosome, is a wrong concept drawing. Okay, so we have to always remember, when we draw the separated sister chromatics, let the centromere face towards the centrosome. Okay, so what are the three labelings we can label here is, okay, I still encourage we focus on the genetic materials, which are many ways we can label here. So this can be called as the separated sister chromatics. But actually the best labelings, the best definition of these drawings is this daughter chromosome. Because now each of the one chromatic here, they are going to become the chromosome for the daughter cells. So these are the daughter chromosomes. Or you can just label, one, this is chromosome, still chromosome, because they are going to become the chromosome of the daughter cells. Okay, so again we have centrosome. Okay, you can mention the spindle fibers, the shortened spindle fibers, or we just say spindle fibers, and you can label the centromere. Okay, always remember, let the centromere face toward the centrosome. Okay, so if we study this diagram carefully, right, now you can notice we still have 4 chromosomes in this cell. Okay, we have 4 chromosomes in these cells. Okay, why? Because we have 4 centromere now. Okay, 1 centromere here, 2, 3, 4. So in anaphase state, right, suddenly the chromosome number become double. Okay, become double because they are get ready for the formation of daughter cells after the telophase and cytokinesis. In the last stage of mitosis, the shape of animal cells show significant changes because telophase will follow immediately by the cytokinesis. So we always draw the cell to indicate the beginning of the cytokinesis, okay, where this is the formation of the cleavage furrow. Okay, a groove that starts to be contracted by the contractile ring of microfilaments from outer inwards. And in the cytoplasm of the cell, the nuclear envelope and nucleus reform. Okay, so I draw the nucleus with dotted technique and the nuclear envelope in the dashed line. So I draw another one here because there are 2 daughter nuclei will be formed. Okay, so this is the nuclear envelope and here is the nucleus. Okay, but in the nucleus, okay, you imagine just now during the anaphase, right? Okay, 2 chromosomes will be pulled to opposite poles of cells. So this nucleus will have 1 chromosome. Each of it have 1 chromatic. Okay, each of it have 1 chromatic. Okay, which represent 1 DNA. Yeah? Okay, so we understand that. Okay, we know that now in the telophase, chromosomes start to decondense, or we say start to uncoil and lengthen, become chromatin. But chromatin, the drawings is too fine, so somehow it's difficult to identify the colors, okay, to study the situation of the chromosome. So since that this is about the usage of color to understand the origin of the chromosome, so I remain here the chromosome in this form, in this pattern. But actually we know that they already become chromatin, okay, the fine fibers. Okay, so same goes to another daughter nuclei. Okay, they also have their chromosome, which have been pulled to the opposite poles of cell and surrounded by the nucleus. Okay, at the opposite poles of cell, the centrosome with the central still acid, okay, masih ada, but now no more microtubules or no more spindle fibers can be found in the cytoplasm because they start to disintegrate. Their mission is finished, complete. Okay, so this is the suggestion of the schematic diagram here to understand or to represent the telophase. Okay, let us draw or let us label, make labeling. So in one cytoplasm now, because the cytokinesis just start with the formation of cleavage furrow, haven't completed yet, but here, okay, in one cytoplasm we have two daughter nuclei. Okay, and in each daughter nuclei we have the chromosome and the reformation of the nucleus, but here is the nucleus. Okay, and also the centrosome with the centros. Okay, so if let's say complete the formation of the cytokinesis, right, they are going to form two daughter cells. Okay, they are going to form two daughter cells after the cytokinesis, and in each daughter cells in the nucleus, okay, they still show the pointy level same as the parent cell, which they are still diploid with two chromosomes. Okay, this is the significance of mitosis. From one parent cell, they're able to produce two daughter cells, and both have the same genetic number, okay, as the parent cell. Okay, if you look at here, this is because, okay, why we still call it as diploid, because the chromosome found in this nucleus, they are same type. Okay, when we study the shape, the size, and the genes carried by this pair of chromosomes, they are 100% similar. Okay, that's why this is diploid, and there are two chromosomes. Okay, usually how we calculate the chromosome, you can count the centromere. So there are two centromeres, so two chromosomes. Same goes to this daughter nuclei. Okay, this is why we can notice that, okay, there are two chromosomes also, and both are same type, so they are diploid with two chromosomes. Okay, this is why in Anaphase, the separation of sister chromatic play important role also, because they divide the set of chromosome equally to the opposite poles of cell. So at the end of cytokinesis, the two daughter cells will inherit the same set of chromosome as the parent cells.